I'd like to thank the meeting organizers for the opportunity to speak a little bit about our program and the development of vaccines for the treatment and prevention of breast cancer. My name is Keith Knutson. I am professor of immunology and cancer biology at the Mayo Clinic in Jacksonville, Florida. It's a pleasure to be here today. A few disclosures: some of the vaccines that we've been preparing, we've licensed those out, and we've been collaborating on conducting clinical trials. So the learning objectives today are to understand the basics of the immune system, what is targeted in breast cancer with respect to vaccines, and how do patients help us decide on the approach that we need to take in order to make vaccines effective, and then have a little bit of an understanding of the regulatory pathways and funding pathways that are available to us to move forward novel vaccine strategies for the treatment and prevention of breast cancer. So before I go into that, I'd like to comment a little bit on the basics of biology. And we all know that your body is composed of many different types of molecules. DNA is an example, RNA, proteins. Proteins are the main machines of your body, and they can have many different properties and carry out many different functions. The reason I bring this up is because I'm going to be referring to fragments of proteins that we use in preparation for a vaccine. So proteins are actually strings of amino acids. So when you string together, for example, 8 amino acids, that is what we call a peptide, and then larger strings of amino acids become proteins. The adaptive immune system in your body is your drug-making machinery. It is a very important arm of the immune system. It's only one arm of the immune system that protects you from infectious diseases and other types of environmental insults that may cause harm to your body, including cancers. The adaptive immune system adapts to various types of antigens, specifically through genetic recombination that occurs in 3 groups of cells, either CD4 T cells, CD8 T cells, or B cells. So CD4 T cells are the central mediators of the immune response, and they coordinate the development of all kinds of different types of immune responses that can impact your ability to fight off an infection, for example. One of the cells that it activates is the CD8 T cells. The CD8 T cells are very important for killing viral infected cells as well as tumor cells. And the last component of the adaptive immune system is B cells. B cells are very important for the development of antibodies, and antibodies have very important roles in preventing the development of an infection or a tumor, and they do this through modifying signaling into the cell. They do this through activating this process called antibody-dependent cellular cytotoxicity, and they activate different components in the blood called complement that can cause damage to whatever is infecting you or a tumor cell and cause it to lyse. 2 things to remember here as we move forward. The B cells, which I already mentioned, produce antibodies. They produce antibodies that float around in the bloodstream, and these antibodies can then bind directly to a tumor antigen. HER2/neu is a great example. HER2/neu is a form of breast cancer that we have been able to treat for many, many years by development of immune-based therapies, notably trastuzumab and pertuzumab, but also the recent development of antibody-dependent conjugates such as TDM1, which is a form of trastuzumab that's conjugated to a small molecule inhibitor of cellular processes. So in contrast to these free floating circulating antibodies that are produced by B cells, T cells, in contrast, recognize antigens that are presented in the context of what we call these MHC molecules that are on the cell surface here. They're often called transplantation antigens. So it's very important to distinguish the difference between B cells and T cells as I get into a little bit more in detail. Try to keep it at a level that you can appreciate. But what's important is that the vaccines that we design are designed to identify these little epitopes that fit into these transplantation antigens that we can pool into a vaccine and then stimulate the body's immune system. Now, there are several different types of immune responses. If you think about it, the environment is full of all kinds of different pathogens, for example, viruses and different kinds of parasites and fungus, etc. One of the unique properties of your immune system is the ability to adapt to these different types of infections, including tumors. And they do that through the differentiation of the immune system down 3 different pathways. Now, I'm not going to go into the unique aspects of each of these pathways, but it's very important to keep that in mind as we move forward. And I'm going to talk a little bit about the types of approaches that we take to develop an immune response in an individual with vaccines. So Th1 and Th17 are 2 types of immunity that we're very much interested in developing for cancer vaccines. Now, this is not unlike infectious disease vaccines at all. In fact, in infectious disease vaccines, really the goal is the same as a cancer vaccine. The unique difference is that with infectious disease vaccines, we try to generate a very strong B cell response so that you have lots of circulating antibodies in the blood that can prevent a viral infection. Whereas in tumors, we generate the T cells that can activate macrophages, which engulf and eat up tumor cells, activate cytotoxic T cells, or these CD8 T cells that I was telling you about that can directly kill tumor cells, and activate other types of cells called monocytes that help further development of the immune response. So again, the goals of an infectious disease vaccine and cancer vaccines are very, very similar. It's how we skew the immune response towards activation of these so-called ancillary cells that are a part of the immune system that's very important, which I'll discuss a little bit more. In terms of the goals of vaccine in the cancer setting, there's 3 ways that we think about the use of vaccines. In infectious diseases, the primary use is to prevent disease occurrence to begin with. And we liken that to COVID, for example, in which the COVID vaccine is given, typically prior to the development or the exposure to the virus so that you have a strong antibody response. And that strong antibody response can prevent infection and help you get better should you be exposed to the virus. In contrast, we use cancer vaccines in the therapeutic setting, in the prevention of relapse setting, and even more recently, as I'll talk about, the primary prevention setting. In the therapeutic setting, the goal is to induce tumor shrinkage. There's one vaccine that's approved for prostate cancer called Provenge, which is given to individuals with metastatic prostate cancer, which has been shown to improve survival. Our laboratory, as well as other laboratories, have been involved in developing vaccines that can prevent relapse after optimal treatment in patients with breast cancer. There's one vaccine that is or has been widely tested and came close to approval in phase 3 clinical trials. That's called Nelipepimut-S. And this is a HER2/neu vaccine, very similar to the vaccines that I'm going to discuss today, and I'll talk a little bit more about this vaccine later. More recently, we've been very much interested in developing vaccines for primary prevention. Now, it's not the first time that vaccines have been used for the primary prevention of cancer. We have 2 vaccines, the hepatitis B virus vaccine and the human papillomavirus vaccine. Both of these are very, very useful in preventing the development of liver cancer and cervical cancer, respectively. but they do so in a different format or a different way than we use vaccines for breast cancer, as I'll get into, in that they block the causative agent of those cancers, the liver cancer or the cervical cancer, by preventing the viral infection, which ultimately causes transformation of the cells that it infects and causes them to become cancers. So there are different types of vaccine targets that are available to us. So I had previously mentioned that your body consists of proteins, and proteins are the main machinery of the body. And that's not completely true. There's carbohydrates, as I had already mentioned, there's nucleic acids or DNA, there's also other forms of RNA and other nucleic acid-based molecules that can have a variety of different functions. There's also lipids or fats that are in the body that can be targeted by the immune system. The vast majority of vaccines target proteins. And there are different types of proteins that we target in cancer. I talked about the HPV or human papillomavirus vaccine that targets microbial antigens. We call them neoantigens because they're new to your body. And as a result, you're expected to generate a broad, robust immune response to those because your body's never seen those before. Now, we know in breast cancer that there's no viruses that cause breast cancer, or at least the vast majority of breast cancers. So the option of developing a vaccine based on a microbe like a virus is not really possible in this particular case. Another form is self-protein derived neoantigens, what we call subdominant epitopes. And so ordinarily, and I'll talk a little bit about this on the next slide, the cell produces proteins at a very conserved level within the body. And your immune system gets used to those levels. And as the expression of certain proteins like HER2/neu, for example, and HER2/neu positive breast cancer increases, that leads to immune recognition. And I'm also going to share our program or information about our program on targeting mutated protein-derived neoantigens. So we know that in cancers and breast cancer as well, that there is sloppiness in terms of reproducing DNA or fixing DNA, and that results in mutations. And your body's immune system can recognize those mutations. And then there's different platforms that we use. Peptides, which are just small fragments of proteins, nucleic acids. Sometimes we use cell-based assays, or vaccines rather, to develop immune responses in individuals. Sometimes we modify viruses or vaccines or bacteria and give those to individuals that can generate immune responses to tumor-specific antigens. So remember, we can develop antibody responses, but that's typically not the approach that we take in cancers, or we can develop T-cell responses. And T-cell responses are generated against these antigens, in our case, protein antigens that are presented on these transplantation antigens. And rather than antibodies, your T-cell recognizes those antigens through unique molecules called the T-cell receptor. I want to go back just for a moment to the foundation of the majority of clinical trials that we're conducting, and that is the development of vaccines for the prevention of recurrence and targeting self-antigens, so antigens that are normally expressed in your body but at very low levels. And just give you an essence of what I mean by this idea as the protein increases in the body, you get very unique parts of the proteins that are presented to the immune system. So in a normal cell with normal levels of HER2/neu, for example, on the top of the cell, fragments of that HER2/neu would be shown. And at normal levels, it's only the dominant regions of the HER2/neu molecule that are going to be presented. And your body's okay with that. They see that, and the immune system says, well, this is really not a problem. We're not going to attack that tissue. That's called tolerance. But as tumor cells increase the expression of certain antigens, they get these very unique epitopes which are presented at the cell surface. And your body can see that. We can train the immune system to see those and to prevent further problems associated with the development of cancer. And we use modern bioinformatics strategies in identifying those regions. And I'm not going to go into too much detail because it's very extensive in terms of how we do this, but bioinformatics are computerized ways of analyzing proteins, and we analyze sequences, and then we conduct a number of different studies in the laboratory to meticulously go through and identify which ones which ones of these epitopes or peptides binds to the transplantation antigens that I was telling you about, and that can be useful for generating immune responses. And we're also helped by what's called natural pre-existent immunity. So natural pre-existent immunity occurs in some individuals. And we go to the patient population, we draw their blood, and we ask them which ones of these peptides have been recognized by the immune system. So all of this together, computational methods with computers, MHC binding or transplantation antigen binding, T-cell activation assays in the culture dish, as well as natural pre-existing immunity, helps us define vaccines that can be potentially useful in patients with breast cancer. This is just an example. We took the HER2/neu molecule and we went through a computational approach to identify 4 peptides. And I labeled those based on where they show up in the protein itself. So one starts at amino acid P59. Another one starts at 88, 422, and 885. And we identified those through those bioinformatic approaches, and they bind to all of these different types of transplantation antigens that are in your body. And then we went to the patient population and we made sure that you generated immune responses, or the patients generated immune responses, and then that validated this as an antigen that we want to include in our vaccine. And then we conduct phase 1, phase 2, and phase 3 clinical trials. So this is the regulatory pathway. So this is the typical pathway that you would use for the development of a drug. So in phase 1, we test a small number of individuals, and we're primarily focused on whether or not the approach that we're taking is going to be safe, because if it's not going to be safe, then we can't use it. And then we go into phase 2 clinical trials where we start to get some idea of whether or not this is a vaccine that can have an impact on the disease. Most of our studies, as I'll get into, have focused on the development of vaccines that can prevent recurrence in individuals with various types of breast cancer. And if that's successful, where we get some kind of signal that it prevents recurrence, then we can go into an advanced phase clinical trial. And that typically is done at multiple centers with over 1,000 individuals. So this can take many, many years to do the whole regulatory process here. It takes up to, perhaps 10 to 15 years to develop a vaccine. So this is a phase 1 clinical trial of that vaccine that I was just telling you about a couple of slides ago, where we identified epitopes that were derived from HER2/neu that are shown here, the 4 epitopes. And you can see that in a phase 1 clinical trial, we could demonstrate that we could immunize individuals with this particular pool of peptide vaccines. Now, we had designed this vaccine so that it was useful in the vast majority of patients. And indeed, not everybody generated immunity to each of the different peptides that were in there, but 100% of the patients developed immunity following vaccination, and that immunity was persistent and it was safe. So it didn't have any impact on normal tissues in the body that perhaps expressed lower levels of HER2/neu. And then we ask in DCIS patients prior to surgery whether or not there was an impact on the disease. And so as you can see here, a very short course of vaccination prior to surgery and routine treatment of DCIS in individuals, you can see here that vaccine caused a significant reduction in tumor size. So the vaccine that we developed for HER2/neu is safe, generates immune responses, has an impact on the disease. And as a result, we've taken that into a phase 2 clinical trial, which has been developed and is ongoing now, and that's funded with the Department of Defense, a phase 2 in which resected HER2/neu positive breast cancer patients, that means those that have, typically stage 2 or stage 3 disease that's been surgically removed, can be vaccinated during the ordinary course of their treatment to stimulate their immune response. So in this particular case, we're very much interested in efficacy, as I told you. So it's very important that we test the vaccine, in this case, along with TDM1, which is, of course, Herceptin combined to a drug, in comparison with individuals that received a placebo. And in fact, we are expected to enroll 240 patients in this particular clinical trial, and about 2/3 of those will be recruited into the HER2/neu vaccine arm. This will allow us to monitor whether or not the vaccine specifically has an impact on disease. And we're very much interested in going into a patient population that's at high risk for relapse. And so the vaccine or placebo will be given to individuals that did not demonstrate a pathologic complete response to standard therapy at the time that they had surgical resection of their tumor. So this is ongoing. If you are interested, I can give you more details. It's opened up, and we're actually at about 20 patients into this particular clinical trial. And similarly, we've done this with other antigens. So that was HER2/neu, where we've carried this through the pathway. This particular one is the folate receptor alpha. And I'm going to really essentially run through this very quickly. It's very similar. We identify epitopes that we see evidence of individuals that have had cancer responding to relative to normal healthy donors. And that really helps us identify those regions of the protein that we can formulate into the vaccine. This is data here showing that patients with breast and ovarian cancer actually have higher levels of immunity to this molecule, which is the folate receptor alpha, which is overexpressed on triple-negative breast cancer and ovarian cancer. So the folate receptor alpha vaccine was developed and published in 2018 in a phase 1 clinical trial, demonstrating that we could take the fragments of the protein, which are shown right here, and generate safely immunity in individuals. And then if we looked out at between 12 and 18 months after we stopped immunization, we could still see that patients had immune response. So that's the ideal outcome of a vaccine for the prevention of recurrence in individuals, is to generate memory immune responses. And that's what we expect with infectious disease vaccine, is to generate immune memory that can come back when you're potentially reinfected with the virus to which the vaccine was targeted. And this has also gone on to a phase 2 clinical trial to evaluate efficacy with Katie Ruddy at the Mayo Clinic in Rochester and Edith Perez at the Mayo Clinic in Jacksonville. So this is a multi-site clinical trial that's going to enroll 290 patients, and 2/3 of those will be randomized to the vaccine as compared to the placebo group. This is data showing that triple-negative breast cancer, specifically relative to ER-positive breast cancer or HER2/neu positive breast cancer, more uniformly expresses the folate receptor alpha and would potentially be a good target for this vaccine. We're actually at about 250 patients that have enrolled in this clinical trial. We expect to be done by June, and we'll have 2 years of follow-up. So this is a phase 2 clinical trial, and we're very excited to see whether or not the generation of immune responses is able to protect triple negative breast cancers from recurring following conventional treatment. Now I want to turn our attention to this other concept that I talked about. So I talked about vaccines that were useful for generating immunity against self-antigens, so overexpressed antigens, HER2/neu, for example, folate receptor alpha. But with modern sequencing strategies now, it's been possible to really start developing personalized vaccines. Because the downside to the HER2/neu vaccine or the downside to the triple negative vaccine is that it only targets a small population of individuals. So there are no really good single antigens that cover the vast majority of breast cancers. In fact, you have to have multiple antigens to cover all of the different breast cancers in one vaccine, which we are doing, and I'm going to get into that. However, as I had mentioned, modern sequencing, modern bioinformatics now has allowed us to sequence tumors. And so this slide depicts again what I showed you. So proteins are made of peptides, and peptides are made of amino acids. So often what happens in a tumor is that there's a change in the genetic code that incorporates a different amino acid into a peptide and a protein, and that allows your immune system to recognize that tumor because it looks foreign relative to normal healthy cells. So that's just an example of two amino acids that got changed. So if your immune system recognizes this peptide, it may not recognize this peptide here because this is a normal peptide that is present on your normal cells. And so with mutation processes, amino acids get changed, and we can identify those changes and formulate those into vaccines. It's very complex. I'm not a bioinformatician myself, but I work with a great team of bioinformatics, and this is the kind of information that they give me. It's very complex. So we have to, I'm obviously not going to go into too much detail about this. It's very complicated, but they use mathematical and computational methods to look at your DNA and see what it looks like. Is it different? How is it different? Where is it different? And then we can find what are the most important differences that we think that the immune system would be able to recognize. So it's a long process. We've started this program, and the idea is that we can use DNA from the tumor as well as your blood to make comparisons, and we can identify those peptides that are unique to the tumor cell. So this is an example of where the peptide fits into this transplantation antigen, and then you have a cytotoxic T cell that recognizes it. So if we can find those, we can formulate those into vaccines. And in fact, this program that we developed, this computational program, was validated and has been shown to identify changes in overall survival. And I'll explain that to you just very briefly here. And so we identified neoantigens, and there's different types of neoantigens, and I'm not going to go into those. And these neoantigens are associated with overall survival. And so individuals with low neoantigens tend to have less or have worse overall survival compared to those individuals that have medium or high levels. And that is important. And it also includes these neoantigens or these mutated derived neoantigens that can bind to what we call class 1 molecules and activate CD8 T cells, or those that can bind to class 2 molecules and activate CD4 T cells. So we've identified a program, a very intense computational program that can interrogate your DNA. It can identify foreign regions of that DNA, which give rise to foreign proteins. And we also validated that this is an approach that we can actually see a difference in overall survival naturally in patients with breast cancer. So operationally, we'd like to take this approach into developing therapeutic vaccines. So how would we use those? We obviously can't use those for prevention setting, because in prevention setting, you don't know what those neoantigens are going to be. But we can use them in therapeutic settings. In fact, our laboratory has been working on improving the response to immune checkpoint blockade, which is used in triple negative breast cancer, Keytruda, for example, but it's not quite as effective as it is in other cancers. But if we use vaccination, we can improve immune checkpoint blockade. This is actually studies from a mouse, and there's this vaccine here, which is called C3L3N1. And this is a vaccine that is composed of mutated neoantigens from in this particular case, a mouse breast cancer. And we can see that we can generate immunity to these peptides that are circulating in the peripheral blood. And if we add anti-PD-1, which is Keytruda, we can boost those immune responses. But importantly, in this particular mouse model, and in most other mouse models of breast cancer, anti-PD-1 doesn't have any therapeutic activity at all. In fact, it doesn't improve survival relative to doing nothing. But if you add in the vaccine along with anti-PD-1, which is shown here, you get greatly prolonged survival. And in fact, 75% of the cancers that we see in mouse models where we treat with anti-PD-1 plus vaccine actually completely regress in these mice. Now, in some cases, they grow back out, and we're studying why they grow back out. There's different types of mechanisms that ultimately result in recurrence. But the main point here is that vaccines improve immune checkpoint blockade, and they can do it safely, and they can prolong the end of the life of these mice, and even potentially patients with metastatic breast cancer. So we're actually using this information, and then we now have approval from the FDA to move forward with a clinical trial. And how this is going to work is shown here. So this is initial clinical trial is going to include not only breast cancer patients, but patients with other cancers as well. So we can really understand the safety and feasibility. So individuals that have metastatic cancers that are being treated with pembro and or chemotherapy that don't respond can ultimately enroll in the clinical trial. And we'll take a sample of their DNA from their tumor. We'll take a sample of the DNA from the blood, and we'll interrogate that with that program that I was telling you about to identify these foreign mutation-derived neoantigens. And that usually takes between 8 and 12 weeks to do all the sequencing and then come across my desk, and then I arrange with the manufacturer to make the vaccine. And so we've actually started this approach as we were recently approved by the FDA. And then once that vaccine is ready, then we treat the individuals with a short low-dose cyclophosphamide treatment and then start the vaccine series. And we do that in combination with pembrolizumab. So the hope here is that patients with advanced metastatic breast cancer, or even unresectable stage 3 breast cancer, can actually go on to pembro plus vaccine, and the hope is that we'll see shrinkage of the tumor that wasn't observed with pembro plus or minus chemotherapy alone prior to the administration of vaccine. So we're very excited about this. It's a very difficult and rigorous pathway to get there, but as we continue to move forward with our phase 1 and phase 2 clinical trials, we'll have a more of an understanding of, are we targeting the right targets, but how feasible it is, and where can we improve the feasibility so that we can get those into patients that have a need for novel, innovative therapies. Now I want to turn our attention to also, how can we decide on the vaccine approach? So there's different types of vaccines. I more or less mentioned this to you. There's vaccines that we can target with parasites. We can target bacteria and fungus that's at mucosal linings, so in the gut or in the lung. We can, target vaccines to generate viral or bacterial-like infections. So there's all these different types of flavors in an immune response. So this is a very complex slide, and I don't want to go into it, but what I want to convey to you is it's important, and this is how we operate in our laboratory, is to identify those strategies that we can generate immune responses in individuals, not by just guessing at what type of immune response, but asking in patients what type of immune response, and this is without vaccination, ordinarily is linked to better outcomes. And so there's been a wide variety of studies in which we can see different flavors of the immune response that's shown here in these boxes. But what we see in individuals with breast cancer, for example, is that those responses that are associated with improved survival are only a few of those subsets. So here's a subset of T cells, here's a subset of T cells. So we can use information now that we can glean from sequencing data from individuals to help us understand what is the best approach for you in order to make that vaccine, and then we can adapt those vaccines. So in addition to the antigens, learning the best immune response, we can combine those tools to come up with personalized approaches to vaccination. So this is just a good example of how natural immunity informs on vaccine. So what we know, and this is in ovarian cancer, is that high Th17, that's one type of T cell, is associated with improved survival in ovarian cancer patients. That's shown here. I mean, markedly improved survival. And it's also associated with reduced immune suppression in the tumor. And so we thought, well, this may be an interesting opportunity to figure out how we can develop vaccines that generate high levels of IL-17 or Th17 type immune response is one of those flavors. And so we crafted a cell-based vaccine and we gave it to individuals with ovarian cancer. And as you can clearly see here, relative to standard of care, in which patients almost uniformly, 90% of them by 1 to 2 years relapse, we actually prevented about relapse in about 40% of those individuals. And they're all still alive today, and they're off of monitoring because they've been essentially cured by developing a personalized vaccine that targets that immune response, which focuses or has potential clinical benefit for their cancer. And this is just other relational data that we collected showing that we could generate immune responses, and these immune responses were durable. So lots of opportunities here with newer technologies to ask what antigens, again, and what type of immune response. So in terms of neoantigens, they may be particularly useful in this kind of approach too. And this is just going back to the neoantigens and talking about the potential use of neoantigens to formulate vaccines for every cancer patients that comes in. One of the important things about neoantigens, important features of neoantigens, is that they're common in patients with breast cancer, but they're generally not shared. And so each patient has an average of 6,282 neoantigens that we can potentially target. And when we looked at the TCGA data set, so this is the data set that was assembled by the National Institutes of Health, we see that 99.75% of the neoantigens that we identify in patients, in that cohort of patients at the NIH, the 875 or 895 patients, 99.75% of those neoantigens are only found in one patient. So this is important because each patient has its own unique antigens, and with the ability now to identify these antigens, the ability now to stimulate the appropriate immune response, the hope is that we can have an impact on the disease in individuals based on a personalized approach. So lastly now, I want to just end with a little bit of discussion about a program that you may or may not have heard of in association with the National Breast Cancer Coalition. In fact, this is a program that was started by the National Breast Cancer Coalition many, many years ago, where we had questioned, and what I mean by we is the advocacy community for the development of new therapies and new means of prevention of breast cancer. Would a prevention vaccine potentially work in this population? So we think about prevention vaccines, we tend to think about prevention vaccines that target viruses that cause cancer. So for example, as I had mentioned, human papillomavirus or the hepatitis B virus. So in the case of breast cancer, there is no, specific virus or bacteria that causes the cancer. So that was out of the question. So we went through a number of different meetings, annual meetings, to try to identify the vaccines. And I'm just going to be very brief about this. This is a work in progress. So the whole goal of this, and it's called the Artemis program, is to develop a vaccine that targets all of the three major subsets that can reduce the incidence of breast cancer, to prevent it from happening in the first place. It's not going to be like a viral vaccine where we're blocking a virus. It's going to be a vaccine in which we stimulate immunity in individuals against HER2/neu, one of the antigens that I talked about, or other antigens. In fact, those antigens are shown on the next slide here. We're going to target HER2/neu, which is amplified in about 20% of cancers. Another one is MAGE-3, MUC1, you may have heard of some of these, survivin, mammoglobin A, and H-TERT. So the biggest challenge is, of course, we have to get all of these into the vaccine. And we have, with funding from the National Breast Cancer Coalition, we've actually done this. So this is very complicated here, but the whole premise of showing you this slide is that we've been able to achieve from a single vaccine expression of all of the different antigens within a small cocktail, so that we can deliver those to individuals. We have a number of different challenges ongoing, despite the fact that we've made the vaccine. Of course, these programs are very expensive to get those into clinical trials, and we've been working now with the National Cancer Institute, who has enabled us to manufacture the vaccine at the high quality level that we need for human studies. And in fact, within the next year or two, we'll be able to test this vaccine first in collaboration with the FDA in stage 3 and stage 4 patients. They're very much interested in this approach. And then we're trying to think how we can move this into a high-risk population. This is a very significant challenge because these high-risk individuals, see BRCA1, BRCA2, or others that have high risk. It's not like COVID or other infectious diseases. Oftentimes, it takes a long time or many, many years for those cancers to develop. So we need to have some way of knowing whether or not we can impact disease prior to going into those high risk individuals. And so our thought is that we can go into very early stage cancer in the neoadjuvant setting, so prior to surgery, and we can ask whether or not the vaccine has an impact on disease. Now, it may not shrink tumor, but does it induce the immune system to recognize the tumor? That's what we're very interested in. And so if we have confidence that the vaccine is safe and effective at generating immunity, and effective at generating immunity that moves into the tumor, then we can think about how to apply this to individuals that are at high risk for developing for breast cancer. So more to come on this. So there's lots of great opportunities. We're very proud to be at Mayo Clinic, to be working with the National Breast Cancer Coalition and now with the National Cancer Institute to advance this concept of a prevention vaccine to stop breast cancer. So in conclusion, vaccines for cancer are used in both the therapeutic and prevention settings. In infectious diseases, they're typically used in prevention settings, and they can target mutated proteins as well as overexpressed proteins like HER2/neu. And they're potentially useful alone, or as I had mentioned, in combination with immune checkpoint blockade. This may be an opportunity to improve the outcomes of immunotherapy in patients with breast cancer. And we know that in a variety of different studies, including ours, there is benefit of these vaccines. The question is, how can we get them to advanced clinical trials and how we can get those approved? And so this is just a meta-analysis of the early phase clinical trials with, I like to call it an E75 vaccine. It's actually one of the first vaccines that I developed, but it's called Nelipepimut S. And that's shown potential efficacy. In fact, it reduces the rate of recurrence about 40%, but it failed phase 3 clinical trials for other reasons. So vaccines work. How to use them and how particularly to advance them into a clinically useful, FDA-approved, licensed manner is a significant challenge, because there's lots of competition with other, types of approaches that are being done for the treatment and prevention of breast cancer. So just to wrap it up, I'd like to acknowledge my colleagues at the Mayo Clinic and the National Breast Cancer Coalition, other colleagues throughout the years, as well as a number of funding agencies for providing support for us to conduct these studies. And with that said, I thank you again for the opportunity to go over our vaccine program. There's other vaccine programs that are being conducted across the United States. And the hope is that one day we'll be able to get one or more vaccines that have an impact on breast cancer, either in the treatment setting or in the primary prevention setting. So I thank you again.